Okay, everybody, this is the second part of Chapter 16 that we need to discuss. I'm starting on page 6 of your notes. You may hear some piano in the background. My son and daughter are playing the piano. But we're going to skip these things for now, and that was more or less how we got to the discovery of DNA being the transforming agent. We're just not going to worry about it right now. We got to kind of move ahead. So we're going to pick up with Watson and Crick. They were the ones who finally put together this model that you see that was anti-parallel and with the nitrogenous bases in the middle of the molecule being held together by hydrogen bonds and all that kind of stuff. Um, and what was the significance of knowing the above structure? Who cares? Well, when they did this, now we understood how the molecule was replicated and how the molecule was read. It was some pretty high-end stuff that we're still picking apart today, some 60 or so years later, basically. So a couple things. First, we talk about DNA replication. All right. So DNA replication, most important, you should know that this occurs during the S phase of the cell cycle. Very important. Okay. It takes a long time. The average cell takes about 12 to 14 hours to do this, which is double the amount of time spent in G1 or G2. You should also know that the method is called the semi-conservative model of replication. And what that means is when DNA is replicated, guys, it starts off as one molecule. It's going to divide and split into two. But the molecule is split right down the center, and each parent strand, each old strand, receives a new daughter strand. And that's what's being shown right here. The one color, the dark blue, represents the original parent strand, and the light blue is going to be the daughter strands, okay? And they figured that out based upon using some isotopes. We're not really going to spend much time here on this, but you should know semi-conservative model means that when DNA is replicated, each side receives one new side, and that's how it works, okay? So it's very hard to illustrate when you're not in class, okay? And then, more importantly, we look at the difference between like eukaryotes and prokaryotes as far as their DNA goes. These are prokaryotes. Prokaryotic DNA is circular. It's a loop of DNA. And it's going to replicate like eukaryotes very quickly, though, okay? It's going to form what's called a replication fork. These are the forks over here. And this is the origin of replication right there in the center. And then is going to replicate going two directions, okay? Going two directions, so that here's the original parent strands, and here's the new daughter strands. So just like eukaryotes, it's going to be semi-conservative. Eukaryotes are linear DNA, more importantly. They're not circular. Chromosomes in your body are linear, okay? And each chromosome in your cell, there's 46 in each one of your cell. Each chromosome is made of one molecule of DNA. So each one of those gets replicated separately. And that's when it forms its sister chromatids during the S phase. So the difference here is instead of forming one replication bubble, you're going to make many replication bubbles. Okay, and those replication bubbles are going to move towards each other until they run into each other, in which case you're going to make two new daughter molecules. So if you look very carefully here, we started off with one molecule right here, double-stranded is what I was trying to say. And when the molecule replicates, each strand has a new daughter strand. Those are the light blue ones right there. That is the semi-conservative model. It's as much as you're going to need to know about that, okay? And here's just some differences between DNA and RNA in prokaryotes and eukaryotes. For example, in prokaryotes, it's naked. It's not wound around histones. In prokaryotes, it's circular. The genes don't contain introns in prokaryotes. We're going to talk about that in Chapter 17, but what that means is that the DNA does not contain non-coding portions, okay? It's all coding. Whereas our genes have introns, which means that when your DNA is transcribed to make a protein, not every single nitrogenous base is actually going to come together and be transcribed into a protein. So we'll talk about that. Internal structures, of course, prokaryotes don't have organelles. Okay, ribosomes, we both have ribosomes. They're just going to be structurally a little different from each other. Reproduction, of course, it can be asexual or sexual. And then average size, of course, prokaryotic DNA is smaller, where eukaryotic DNA is larger. We have more genes, okay? All right, so DNA replication, this is really what we need to look at. Okay, so how it happens, all right? So this illustration is going to be the most important thing to look at. Note we're looking at one piece of DNA that's being made into two. Okay, so here's the original parent strands, as they're called, the dark blue. All right, and this is what's called the replication bubble has been opened up. So if you look right here, here's my replication bubble. So it's going to start right here, and this is called the replication fork right there. Okay, and that's what's going to keep on. It's like a zipper that you're unzipping or on your jacket, where it just keeps on going. The fork keeps on opening. Okay, so there are two sides of the molecule that are going to be made. The new strand that's built towards the replication fork is called the leading strand. So the leading strand gets built towards the replication fork as one big piece. And then there's a lagging strand. The lagging strand is the one that is going to be built away from the replication fork in pieces, and those pieces are called Okazaki fragments. Okay, so this molecule, guys, is built by a series of enzymes. Okay, the first enzyme that comes along is called helicase. Helicase, and its job, thus its name, is to unwind the helix. Okay, it actually unwinds and rewinds. So first thing is helicase comes along, unwinds it. All right, and it also is going to open up the molecule here. Okay, next comes something called primase. Now primase isn't shown right here. All right, but primase is going to come by, and it's going to start laying down a few nucleotides, 5 or 10 nucleotides, and all enzymes, guys, this is super, super important. I don't care what enzyme you're dealing with, even in transcription and translation and replication, they all move in one direction. Okay, just like the boy band. That's why, I sing that song to you guys. I was going to sing that song to you guys, the story of my life. It's all about DNA and all that kind of stuff. I'm telling you, it's deep, all right? But I can't sing it right now. But Mr. Velada and I were going to do a duo. It was going to be really, really fun. So I hope a few of you are laughing right now, because it was one of the highlights of my entire schooling career, teaching career and schooling career. So here's how it works. As you'll notice right here, this old strand right here goes 5' to 3'. That means the new strand right here would be 5', and it's going to be built. All molecules move in one direction. They build the new molecule in the 5' to 3' direction. So that would be the 3' end right there. That's the 5'. So 5' to 3'. Okay. So as I said, going backwards, first thing that comes along is a molecule called, or an enzyme called primase. And primase is going to lay down about 5 to 10 nucleotides. And those 5 to 10 nucleotides, guys, are RNA nucleotides. Okay. So it goes 5', and then it stops, and that's the 3' end. And that 3' end is very important, because the next enzyme that comes in is called DNA polymerase 3. And DNA polymerase 3 needs a 3' end to attach to begin building nucleotides. All right, that's just how it works. So then comes DNA polymerase 3, and it just keeps on building all the way. Now, those original RNA molecules right there, RNA nucleotides, have to be converted back to DNA. So another enzyme comes along and converts them, and its name is DNA polymerase 1. Notice the names of these enzymes. Polymerase, DNA polymerase. It's building a polymer. Okay, many nucleotides. That's all. No big deal. Oh, there goes my son playing the piano again. Okay, this is called the lagging strand, because what's happening is the fork is opening this way, and the lagging strand is the strand that's kind of built going away from the replication fork. So it's kind of going against the grain, if you will. So once again, here's the 3' end of the original, and there's the 5' end of the original. These molecules, these enzymes, can only build in the 5' to 3' direction. So it's got to be built this way. All right. And same thing. Here comes primase. Primase lays down that primer of some RNA nucleotides. It goes 5' to 3', and then it stops at 5 to 10. All right. And then after it does that, DNA polymerase 3 will add some nucleotides to that, usually about 100 or 200. Okay. But that replication fork is moving, so it's kind of going against the grain. And then it stops, and then primase comes in, does the same thing, adds that primer, stops at 3'. DNA polymerase 3 needs that 3' end to attach those nucleotides to, and it goes again. Okay, so you're going to build it in fragments. And polymerase 1, DNA poly 1, is going to convert those RNA to DNA. That red there represents RNA nucleotides, in case you haven't guessed. Okay, and ligase is the last enzyme, and what it does is it links together the Okazaki fragments. Okay, it's going to form covalent bonds, phosphodiester bonds, between the backbone of that molecule right there. All right, this is a little tough, but it's something that you just have to be, as long as you kind of go over it, you're going to be good to go. All right, so these are the enzymes involved, and so helicase is going to unwind and open the molecule. All right, and then topoisomerase, what that's going to do is that is going to kind of relieve some strain on the molecule. If you think about it, you're opening up that molecule. So because you're opening up that molecule right here, there's some tension on the molecule ahead of the replication fork. So it just kind of relieves the strain on the molecule. It's like Advil for DNA. That's a good joke. Single-strand binding proteins, those are really cool. SSBPs, as us nerds like to call them, and you can see it right here. Single-strand binding proteins. We opened up the molecule, but these are sticky ends right here, and they just come right back together. All right, we don't want that to happen. So single-strand binding proteins, what they do is they bind to the molecule, and it prevents the DNA molecule from coming back together. Okay, so single-strand binding proteins, they basically hold the molecule apart. Okay, or SSBPs. That's my next license plate. Primase, of course, it puts down those RNA molecules, RNA nucleotides, excuse me. And remember that it's going to start. It's going to leave a 3' end. Leave a 3' end for poly 3 to attach to. That's the most important. Okay. DNA poly 3, that's the big one right there. And that's going to build the new daughter side. One thing you should know about DNA polymerase is it's a really, really, really exact enzyme. It makes very, very few mistakes. Okay. RNA polymerase, not so much. All right. It does make mistakes. And DNA polymerase 1 converts RNA to DNA. Okay, so that's going to be your replication. This is just some disorder right here, a disease. Thymine dimers, they form usually from UV light, and you can see the thymines they're forming a dimer. They're kind of pairing together. Not good. They form a blip in the molecule. Now it's broken. So these nuclease enzymes, and those are proteins made by tumor suppressor genes, guys. P53. Okay, what they do is they come around, they cut it out, and then poly 3 comes in and repairs it. And there are some diseases out there where people don't have nuclease enzymes, or their nuclease enzymes don't work. Retinosa pigmentosa and xerodermata. I can't remember the last thing, but they can cause skin cancer and eye damage. Okay. Last thing are telomeres. Okay, telomeres are going to be the end of a chromosome. And what happens is, as you get older, with each replication, your chromosomes get shorter and shorter and shorter. They used to think that this was caused aging. And so this is kind of showing you what's happening. All right, so this is what we're looking at with the telomere. So you can see here's the lagging strand right here. All right, and the lagging strand is being built 5' to 3'. And that's the RNA primer. So it goes 5' and it stops 3' right there. And this is the very end. But what ends up happening, guys, is that once that RNA primer falls off, okay, nothing can, DNA polymerase 3 or 1 cannot replace that with RNA nucleotides, because the enzymes only move in one direction, 5' to 3'. So just like it says right here, primer removed, but cannot be replaced with DNA because no 3' end is available for DNA polymerase. Okay, as you can see, it goes 5' to 3'. Here's the 5' end where polymerase, of course, DNA polymerase 3 attached to and began to build the nucleotides. But there's no other 3' end. So what ends up happening is that this side doesn't get replicated, and with each round of replication, the chromosome just gets shorter, shorter, and shorter. And those are called telomeres. So all a telomere is, guys, is the end of a chromosome. Okay, and the telomeres, they're going to get shorter and shorter and shorter. Telomerase is an enzyme that can actually replicate the telomeres. It is not active, though, in somatic cells. It's only active in usually gamete cells, and it's also one of the ways they find cancer. Sometimes cancer cells will replicate their telomeres because they activate telomerase enzyme. And what telomerase enzyme is a special enzyme that doesn't need that 3' end available, and it just puts that down. So that's one of the ways that can actually find cancer, from what I'm told. Very interesting. Just know what a telomere is, the very end of a chromosome, and I'm happy with that. Okay, so now we've kind of finished up chapter 16, and I've left a review for you that I want you to do. And please try to use the Kami, and I've left a code for you in there. You may have to just download Kami. Okay, thank you very much.